sorry, I forget my notebook here. Okay. Yeah. Do you hear me well there, please? Hello?
Yes, we can hear you well.
Yeah, that's what I want to know. All right, just give me 1 second here. I have to close this down so that you don't have the echo. Okay, so, just today, hopefully, I will finalize the zygomycetes. Okay. Uh, the last thing I think I talked with you with the class zygomycete here. Okay. I was talking in the order Glomialis, which is basically include those species in which mostly doing the mycorrhizae. Okay. And the last thing I mentioned to you in this regard, that basically that they are extremely important. Already I mentioned that to you. They are extremely important because they are highly enter the living roots cell of perhaps 90% of all higher plants and establish with them obligate mutualistic symbiosis called arbosicular mycorrhizae or endomycorrhizae. Okay, and I think I showed you here basically that this group here, this is basically what you can see here is already, I think I showed to you that you can see here, this is a cross section and a root. And basically, of course, as I mentioned, mycorrhizae or these fungi in which they have association with the root, there are two types. Either they go inside the root cell, especially of the cortex, and these known as endomycorrhizae, or we have the one which only they will live fungi outside of the root, and these known as ectomycorrhizae. Okay, mention this one here. Now to continue after that, okay, to mention, now these fungi, okay, evolved interesting features. These fungi evolved interesting features. Of course, that enable them okay, to transfer a nutrient to transfer nutrients into the roots. Okay, into the root, and also to store, as I mentioned, to reserve food. Okay. Now, what I try to show it to you here, this is basically, as I said to you here, that this is what you call, these fungi are called arbosicular mycorrhizae, okay? Because these are basically, they go inside the cell and they form what you call like a swollen head like this one, okay? So basically, if we enlarge a cell like this, it's going to like this. This is inside the cell, okay? Inside the cell, how it look like. So basically, like to mention to you here, okay? So now, to explain this phenomenon here, okay? So to mention this regard that most species, means most of these fungi species, which is inside the root, okay, form enlarged, thick walled structure. Okay, thick walled structures, of course, within, as I mentioned, the cortical cells of the cortex, called cortical cells of the root. Okay, cortical cells of the root, and these basically, that's why refer to okay as vesicles. As vesicles. Now, as already I showed you here, okay, and the fungus penetrate, okay, and the fungus, okay, penetrate, okay, penetrates the cortical cells, okay, and becoming highly branched. And becoming, as I mentioned, highly branched inside the cell, okay, forming a feathery lobes, branched, forming, okay, feathery, sorry, feathery lobes, and these feathery lobes are known as arbosicular. Okay. And here, of course, or hence, as a matter of fact, okay, we call them hence, okay, the name, or we call the name as vesicular, okay, arbosicular. Okay, and here we call them vesicular, arbosicular. Now, one thing, of course, going to mention here that there is a difference, as a matter of fact, it's going to occur between the cells in which there are no fungus and some of the cells, all the cells are the same, but there's only one difference in the storing of the starch, and that's what I'm going to mention to you here. between the cell, which is already the fungus in it. Okay, the one to mention to you, to mention this point here. Now, colonized cortical cells differ from adjacent and colonized. From the adjacent and colonized cells only, and their ability of course to store starch. Okay. Now, the last thing I want to mention to you basically that, I'm going to just talk about it here. These are the cell here. Now, these fungi which is inside, they are hyphae, usually non-septate. So the fungus which is inside their hyphae is non-septate. And of course, at the same time, as they go inside the root, also they go inside the cell, and to mention to you. So these fungi, their hyphae usually non-septate. And of course, they spread through the soil and enter living roots. And sometimes, of course, there are different species of these fungi, which is, of course, this group here, mostly of the, what you call arbosicular, these are the endomycorrhizae. And of course, we have different species of endomycorrhizae and different species of ectomycorrhizae. So what I try to point out here, that these fungi, their hyphae usually non-septate and spread through the soil and enter living root. Now, just one thing I'll mention to you. You know, the living root, basically the area in which they enter to the living root, because you know the root, it has the root hairs. So there is most probably they go from inside the root hair, which they go inside, However, as I mentioned, separate through the soil and enter the living root. Now, as I mentioned, in this case where they develop structures, there are different, where they develop structure, structure that are diagnostic to the order. That means that structure we can, Of course, later on when I finish all these fungi, I'm going to go on more detail about the mycorrhizae and the different group and which species, and I'm going to deal with that more, But at the moment, this is going to mention to you here, where they develop structures that are diagnostic to the order. Okay. Now, basically, we finish this group here. Now, going to finish, as I mentioned to you, basically in the phylum, as I mentioned to you, in the phylum Zygomycota, we had the class already. We discussed the class Zygomycete with its order, which is more important. We finish all this. Then we have the other class we'll talk about a little bit here, known as trichomycetes. Okay. Now, this group basically, they are a very large group of fungi, but because when you said they are not economically important, they don't cause much disease or so on, that's why you find less interest in that, because most of them basically, they live within the arthropods and they live like a communal with them. So there is no, they don't do any harm, and that's why, but at least they. So to mention this point here. Now, with regards to the class Zygomycete, these fungi live exclusively attached. These fungi live exclusively attached to the lining of the guts, okay, to the lining to the guts of living arthropods. Okay. And as I mentioned, they do not seem to be parasitic. Okay. And I mentioned they do not seem to be parasitic. Okay. And they are regarded as commensals. They are regarded as, put it down here, commensals. Okay. Simply commensals you can put between two brackets when two organisms they live together without causing each one harm to the other. So commensal basically there are two organisms living together without either harming the other. Okay. Without either harming the other, but at least one is getting benefit from the other. You see, these are known as commensals. So commensal, when two organisms living together, nobody is harming each other, but at least one getting some benefit from the other. Okay. Usually, these fungi they don't have what you call a very large kind of biocelia. To mention this point here, that trichomycetes, okay, do not usually produce, okay, an extensive mycelium. I don't know if you can see this one here really. This is, yeah. This is some of them, how they look like inside the cell. You see, they are like this one here, okay? So they don't have, and most of the time, what you call going to mention to you, they have a form like of a hold fast to hold to the substrate, okay? And also seems most of the time going to mention that when they reproduce, mostly they produce asexually. So even when we try to group them, we can group them only on these asexual type of spore they produce. Okay, so basically these are the fungi here, as I mentioned to you, they are do not produce what you call usually an extensive mycelium. And as I mentioned to you here, that the somatic, okay, the somatic stage is okay of some species. The somatic stage of just species, okay, is confined okay, to a food shell, we call it to a food shell, or hold fast, or hold fast, which is to attach itself, okay? And from this, basically, we had from which, okay, I mentioned from which, of course, a sporangium grows. Okay. As I mentioned to you also, they have limited filamentous branch. They have limited, okay, filamentous. They have limited filamentous branches, okay? Or sometimes they have unbranched mycelium, or they have unbranched mycelium, okay? And the hyphae or the cell, it is multinucleate or cenocytic, to mention to you that, okay? That to mention that upon the hyphae is, okay, uh, multinucleate or we call it cenocytic. Okay. Though sometimes, or some species, though some species are septate. Okay. Before that, those are septate. Now, in this class, of course, we have orders, as I mentioned to you, but most of these orders based on the asexual type of reproduction. I'm going to mention to you here. Okay. There are three orders. If you can put this down here so that you can finish this one. There are three orders in the class Trichomycetes. I'm going to mention down to you here. Okay. There are three orders in the class Trichomycete. And as I said, mostly this is based on their either morphology or the non-sexual spores. Okay. As I mentioned to you, there are three classes. Oh, sorry, three orders in Trichomycetes. And these order basically based, means their classification based, okay, on morphology and we call it non-sexual reproductive structure. Okay. We have one order known as Assyrialis. And we have another order known as Exerinialis. And we have a third order known as Harpelialis. Okay, so these are the three order here. Just to give you a few line on these, because as I mentioned, okay. Now, these basically the first order, which is the Asterialis, these are basically, okay, produce, okay, arthrospores. We'll see that later, okay. Produce produce arthrospores. And as I mentioned that arthrospores are found, okay, or when the thallus, okay, form or forms fragments into spore-like segment. Usually in arthrospore, when there is the hyphae, like this hyphae, okay, and what happened here, the hyphae sometimes will form segment like this, okay, and usually do that when the condition become unfavorable and these segment basically become thick and it can separate. So basically the same cell like this, and when it comes like this, these cells known as, okay, arthrospores, okay, because they come thick and in the end they will pass what you call the unfavorable condition in that what you call protective cell, okay, which you call in that case arthrospores, and they can germinate into a new hive. Okay. Now, with regard to the second order, the characteristic of the second order here, now this group here also they form this group of fungi, basically of, as I said, all based on morphology or non-sexual spores. So basically this group of order, their hyphae forms, okay, branched, okay, cenocytic thallus, thallus, okay, it's basically which cleaves into sporangia. Cleaves into sporangia. Okay. And simply going to mention to you that each sporangium and each sporangium, as a matter of fact, form a single sporangiophore. Okay. And each sporangium simply will form each sporangium, okay, forms a single, means only a single spore, or we call sporangiospore. Okay, form a single sporangiospore, and when it forms that, basically it's going to escape, okay, which then escapes. A single sporangiospores, which then escape, means will leave the sporangium. Okay, so this is with regard to the group, as a matter of fact, of what you call the characteristic of this group of the order Excelliaris. Now, the third order, which is known as Harpalialis, this one here, this order, Harpalialis, Okay. Usually this will go basically from the spore, from the, from a sporangio spore, they will form, as a matter of fact, here several filament, several filament, and disseminated, and later on, and disseminated. Okay. Most of the time, because as you told you, they live in the guts of the what you call of the most arthropod. So basically, when the insect or when the arthropod die or decay, then in that case, of course, the spores are liberated. So basically, this is what all I want to talk about the trichomycetes, though it is a very large order, but as a matter of fact, because they don't causing any much harm, so you don't have much. Okay, this is with regard to this. So now, once you finish this one, I'm going to move to another group of fungi, okay, which is quite a large group of fungi, okay. By this, I will end up what you call all the group of the phylum Zygomycota, okay. Now going to move to this group known as here, going to explain gradually, okay, that the phylum Dicaryomycota. And this basically include quite a bit two large groups, okay. We have in this group subphylum, okay, known as Ascomycotina, okay, and both the class in this one here and the class in this one here, known as Ascomycetes, okay, and the other subphylum, there are two subphylum in this group here. I'm going to just to talk about it a little bit here. The other subphylum is Basidiomycotina, and the class in this one here known as what you call okay, known as Basidiomycetes. Now, the reason we put these are more or less one of the largest group of fungi. We know basically Ascomycetes and Basidiomycetes. Okay. Now, these fungi basically, what I try to point out here, why I call them in dikaryotemycota. This means when they have a cell of two nuclei. Now, as you know, just give me a second here to explain this one, and then I will give you the. Now, though this is here, usually, can you see this one here? Okay. Now, these fungi, when the sexual reproduction will take place, okay, and of course, when the two cells, they are, of course, these fungi basically are either septate mostly, okay. So when the two filament they come together, I don't know if I have another one which is maybe better than this one here. Let's do. Yeah. When you look to this section here, okay, I'm just going. Now, this, for example, it shows to you a mycelia one. As you can see, the cells are almost green or bluish, and these are in which the cell, as a matter of fact, nucleus is red. So basically, when two nuclei as a heterophilous or semi-informed gamete, they come together, okay? And of course, what happened here, then they get a stage in which, as you know, in all the sexual reproduction, what you have plasmogamy, okay? And then you end up with karyogamy. Plasmogamy, in which that stage, as a matter of fact, in which the two cytoplasm unite, but remain the two nuclei still separate. You got this point here now. The other fungi, when what happened is that most of the time after the plasmogamy, most of the time the karyogamy will follow a quite what you call soon or quickly. Okay. Now, in these fungi, what happened here? When the plasmogamy occur and the stage of the two nuclei become what you call, as you can see, this one here, okay, you find cells, as a matter of fact, bluish and the green, okay. Now, the karyogamy, like this one here, will last for a long period, could be sometimes a year or more, and later on, when the two nuclei will unite and produce what you call the zygote and the meiosis. Do you follow this point here? And that's why when that stage of the cell with two nuclei means dikaryon, means two nuclei. And that's why you put this group of fungi in one underphylum called what you call dikaryommycota, because of the length of the period in which the two nuclei phase will last. You follow this point here? Now, when the two nuclei will unite and the karyogamy, and then meiosis will occur, and then they produce what you call their spores. Now, in case of in case of the ascomycetes, okay, I'm going to explain this one here to here, okay. In case of the ascomycetes, when you have the, say, this is two cells here, of the cells here, and the two cells, of course, in which we call that as the dikaryon, okay. One thing I forget to mention to you. Now, usually when it happened that stage of the two nuclei dikaryon, also they produce more cells or hyphae also with two dikaryon. So that's why it will last for a long, and also they produce more hyphae still containing two nuclei, and that's we call all this group, we put them what you call in the phylum dikaryommycota. But what happened here, in the case when later on when the two in ascomycete, okay, when the two nuclei become what you call the karyogamy, and then meiosis will occur. In ascomycete, meiosis is going to occur, they're forming like a sac-like structure, a sac structure like this, and inside that structure, will after meiosis, will develop the spores, okay? And usually this sac-like structure, which is basically known as ascus, and then these spores known as ascospores. You follow this point here, okay? And that's why all the fungi in which after what you call karyogamy and meiosis will develop that sac-like structure, we put them all in one group known as ascomycete, and it is in a very large group. You follow this point here? Now, when they develop that ascus, then later on, they develop like a fruiting body around that ascus, and we're going to classify based on that type of fruiting body. Now, this is with regard what you call to the group at the beginning to tell you what you call known as the ascomycete. The other group, okay, in which sometimes when the two nuclei, okay, after a period of long period, and they unite, becoming what you call karyogamy and zygote. So what happened here? Usually the cell in which they develop what you call like a small septa like this, okay, after meiosis, and then what you call of course, if you have meiosis 1 and meiosis 2, is going to result 4 spores. Now, in this one here, what is going to happen? This cell is going to make like a small structure like this, okay? And each of the 4 spores is going to balloon out like this. You follow this point here? Okay, and that's why what happened in these fungi, because this cell in which happened, what you call it, has the chirogamy and meiosis occur into it, as then that goes, this is called as a basidia or basidium, like a base, okay? And then these spores known as basidiospores. Okay, so basically all those fungi in which they form that basidium with the basidiospore, we put them in one class known as the basidiomycetes. Okay, so that's basically how we divide the two groups here. Example of the basidiomycetes, for example, all the mushroom, including the one you eat, they are all belong to this group of the basidiomycete. Okay, because the mushroom, though I don't have a picture with it, when you have the mushroom, you didn't notice that. Okay, if you have a mushroom like this is the mushroom here. Okay, if you cut that mushroom, okay, and you tap it on a piece of paper, you find it's going to full of spores. So basically, if you enlarge it, because really the mushroom is nothing more than a very tight hyphae, very tight, compact together. It looks like a tissue. That's what the mushroom. But if you come in the microscope and you see underneath it, going to find underneath this one, all of it is cell like this. And each one, basically, it has the basidia like that. You follow my point of view here? So that's why all the mushroom and other group fungi, but all the mushroom belong to the group of the basidiomycete. So basically, once I explained to you here, that is why we call them the dikaryommycota, and why we have the group called ascomycete and basidiomycete, because it depends after meiosis which form is going to develop the cell in which the spores are coming out after meiosis. So now I'm going to deal first with the, of course, I will deal first with the ascomycete, the phylum, dikaryommycota, and I have here, of course, the class ascomycetes, and that's we call them because the common name for them is sac fungi, because they're forming that sac. That is the common name for them, the sac fungi. Okay, and as I showed you here, okay, when they do the meiosis, that's how they do it. For example, to show it to you here, that's how they do their ascus. You see that? And inside, of course, what happened here, because meiosis, it gives you 4 spores, but after that, another division will occur as mitosis, and they produce typically 8 spores, though it shows variation, and that's why we call this is ascus, and this is spore, which is after known as ascospores. Okay. First of all, I'm going to have what you call as a general characteristic of the group here, of all the group that's good. Okay. Okay, general characteristic here to mention to you here. Okay. In general, to mention basically the hyphae of the phylum Dikaryommycota. The hyphae of the phylum Dikaryommycota. In general, of course, and Dikaryommycota, of course, include both the ascomycetes and the basidiomycetes, okay, are narrower. They are narrower. The hyphae is more narrower than all those of zygomycetes. okay, are narrower, okay, than those of zygomycetes. Okay, and basically, sometimes when these hyphae aggregate, they can form a very long hyphae. It could reach sometimes several kilometers. Okay, to mention to you here now, but in aggregate, but okay, in aggregate, okay, they are very long. As I mentioned, they are very long, and sometimes, okay, several kilometers, of course, in length, if you add them together, per gram of soil. Okay. Now, if you remember when you mentioned in zygomycete, you mentioned that zygomycete, the hyphae, the cell is synocytic. Now, in this one here, basically, no, the hyphae are septate. Mention this point here, other characteristic. Now, their hyphae are septate. Okay. And of course, when I septate like this, okay, and of course, each one of these known as what you call septa, the cross that known as septa, okay.
When you say that their hyphae are septate, are you referring to
Yes,
just
they are
the
septate.
class?
Yeah. No, no,
Are
the
you
whole,
referring to just the class Ascomycetes or the entire phylum
all,
Dicaryomycota?
no, no, all of the what you call, all of the what you call of the basidiomycete, all of the what you call dicaryomycota.
Okay, thank you.
Yeah. Okay. Good. Now, the other point I'm going to mention to you here, basically, this group of fungi in general also, they can grow on a wide range of conditions. They can grow in a wide range of conditions. Okay. Now, as I mentioned to you also, that mostly the basically now ascomycetes, as a general here, ascomycetes, okay, can often exploit, sorry, can often exploit, okay, cellulose, okay, and many basidiomycetes some species, of course, in addition to cellulose, okay, to mention, and many basidiomycetes can digest both, depending on the species, of course, can digest both cellulose and lignin, okay, and lignin, okay. Now, also to mention that there are imported fungi, of course, this one here. Now, and to mention also that these species or these fungi, also these fungi in both of them, okay, means these fungi, to mention other of their characteristic as a matter of fact, okay, it can use many different form of combined nitrogen, okay. This one can use, okay, many different forms, okay, of combined nitrogen. Okay, to mention this point here. And also to mention even some group of ascomycetes, as a matter of fact, even they can digest the keratin. Okay, to mention this point here. And okay, and some ascomycetes, okay, even specialized, okay, in metabolizing, okay, keratin. That means when you say like this, of course, that means able to, to decompose it. Okay. Now, also to mention, of course, just to add to you here, to add in on, when I talk a little bit about the zygomycete, about the order Glomialis, okay, in which they have mycorrhizae, also going to find quite a few species of basidiomycetes and a few ascomycetes, also they form mycorrhizae. But when I'm going to talk about mycorrhizae, we can find differences, especially species, because mycorrhizae sometimes, even when they come to the air, they have a primary and secondary, how they develop within the ecosystem. But at the moment, I just want to mention this point here. Now, thousands, the other point, thousands of basidiomycetes, okay, as I mentioned, thousands of basidiomycetes and quite a few ascomycetes. And fair question, okay, form mycorrhizae. Of course, they form mycorrhizae with the higher plant, okay? For mycorrhizae, of course, especially with the trees, especially with tree species, okay? Three, especially like conifers also, like also like conifers, okay? The other characteristic I mentioned to you basically, now this group of fungi, their cell wall is also contain chitin. So I'm going to move now here. Okay, I don't know if you just want it or not here. Okay, so simply I'm going to mention to you they contain chitinous cell wall. Okay. Now, of course, to mention, of course, about the characteristic, which already I talked about it, in which basically that stage of the plasmochemy or dikaryon will last for a long time. To mention this one point, okay? Now, to mention in this regard here, now, the unique occur in their life cycle Okay. And their life cycle. Okay. As I mentioned to you here, a phenomenon. Okay. Which is known or called, as I told you, which is known as the dikaryon, which is simply, it means two nuclei, known as the dikaryon. And as I explained to you, I'm going to put it down for you here. After a sexually compatible nuclei, okay, after a sexually compatible nuclei, uh, form what you call the dikaryon stage or dikaryon phase, which is basically in this case, of course, this is the plasmogamy, that before the nucleus, okay? Which is the plasmogamy here. Okay. As I'm going to mention to you here now, these nuclei or the nuclei, just mention with the nuclei, okay, do not fuse immediately after the pairing of the nuclei. Do not fuse immediately, okay? They do not fuse immediately, of course, to form a diploid zygote. For that, okay?
Oh, Professor, could you adjust your page for us, please?
Pardon? I already mentioned to you here, okay? A diachron phase in which the nuclei do not fuse immediately to form a diploid zygote because they stay for a while. You got this point here. That's clear? Okay? And as you instead, of course, what happened here, even when they form, say, uh, because if you have, this is one nucleus like this, and as I explained to you, and you have another, okay, and you have this, say, plus, and you have the other cell, basically, I just put it like empty so that you can see the difference here, okay? Now, when these two hyphae, they become compatible in the plasmogamy like this, okay, like that, just like that. They stay for a while, and also they produce more hyphae with two nuclei. That's what I'm going to mention to you. And instead, okay, and instead they go dividing. And instead they go dividing, okay, to produce or to populate, okay, more diacritic hyphae. Okay, more diacritic hyphae. Okay. Now, as I mentioned, of course, in case, okay, of ascomycetes, okay, case of ascomycete, okay. To mention here, of course, that the meiosis of the zygote, okay, results, okay, in a cylindrical or sac-like structure, which is known as ascus. Ascus, okay. The meiosis of the zygote result in a cylindrical or sac-like structure containing okay, typically eight haploid spores. Okay. Now, what I'm going to mention to you here, of course, as I mentioned to you that when meiosis, say, in the cell, this I'm talking about now, ascomycete, when meiosis occur, this is the zygote here. We're going to produce an ascus like this, and inside it, typically eight. So basically, this is ascus, and these known as ascospores. Now, this is with regard, of course, we're going to talk about the basidiomycete at the end, but this is where it's called in the ascus. Now, in this group, basically, in their asexual production, mostly they produce a smaller spore known as conidia or body. And to mention this point here, and this group here, let's go ahead. Asexual production, okay, is by budding. But of course, if some of the organisms are unicellular, called budding. Sorry about this here. By budding, that's if it's unicellular organism, or forming conidia. Okay, and as you just give me a second here so that give me a second here now. For example, in case, of course, the two large family group of Penicillium and Aspergillus are this group. Now, normally, what you call, I'm going to show you here for your what you call any for your labeling, if you know it. For example, when the hyphae, then you produce that erect. So in this case, this hyphae was holding the conidia known as conidiophore, and these are the conidia in form of this is in case, of course, of what you call of penicillium. Okay, now. And also, basically, if you want to continue now because if this is the conidiophore, because as you can see, they have one cell like this, and then developing the conidia. Now, this cell here known as strigmata. You see, So you have conidiophore. Don't worry about this one. These are branches here, conidiophore, and then producing a row of one cell of stigmata or two cells, depending on the species. And then, of course, they produce the conidia in a form of a chain. Okay? So basically, that most of the asexual production, of course, that varies from species to species. Okay? This is penicillium. Other species could be one cell, one conidia or two conidia, or just depend, or could be branch or unbranched. But just to let you know that the asexual production in ascomycete, and even we're going to find in some basidiomycete, but in ascomycete, basically by the producing of the conidia on conidiophore, just to mention to you here. Okay? I'm going to show all that again here, okay, but just at the moment. Okay. So as I mentioned, yes. All right. Yeah, assimilation by budding or forming conidia. So I'll explain that to you here. Okay. Now, in general, basically, we call the sac fungi or the ascomycete because by the number of their species, they are one of the largest group of fungi. Now, to mention here, the sac fungi, okay, or ascomycetes, okay, are the largest are the largest of all fungal groups. Okay. The other thing, of course, that most ascomycetes are what you call predominantly saprobes. Most of the ascomycetes are predominantly or mostly saprobes. When you said saprobes, means they are saprophytes, because when you have a fungus, as you know, I mentioned that before, if a fungus basically what you call living on a dead organism, they are saprophyte, and these are the important in decaying. Okay, but if you have a fungus basically lives or need a living host, in that case is a parasite, and these either going to be mutualistic or is going to form a disease. You got my point of view? So there are saprobes, means saprophyte. Okay? Saprobes on plants or animal remains. Okay? For that, but of course, as I mentioned to you here, also we have some, and continue after this one, of course, some are parasitic on plants and animal, and in this case, of course, causing disease. Okay. Well, I think I left about, 4 minutes left. Just a minute here before it's closed down here. Okay. Subtitle before I will try to explain this to you here. Seems I'm running out of paper here. Okay. Now, types. I'm going to see titles here. Just a minute here. Types of ascoma. That these are the fruiting bodies. I'm going to explain it maybe in ascomycetes. Okay, now, you remember when I talked to you basically when the meiosis occur because, and this is of course a sexual production, and they produce what you call ascus like this one here, just yeah, produce ascus here, okay, with the ascospores. Now, it depends, as a matter of fact, later on, they can develop what you call a fruiting body. So there are 4 fruiting bodies are produced by the ascomycete. You can now, either one is known as clistothecium. Do you see it here? Do you see it? They produce clistothecium if it is circular and inside of the ascus. Okay? Or they produce what they call like a flask shape known as a parathecium, or a cup shape known as apothecium, or without any fruiting body known as naked. So basically they produce either clistothecium, I call it clistothecium because they're plural, or parathecia, or apothecia, or naked as psi, or naked. Okay? Now, clistothecia, of course, as I mentioned to you, I'm going to continue next lecture because I think within a minute this is going to shut down here. Okay? The clistothecium is a circular. The apothecium is a flask shape with an opening. I want to explain it here. Apothecium, it is like a scissor shape or naked ascus. Okay, so these are basically the most common one. Of course, there is later on I'm going to add to that. Basically, these are the 4 type of, because we're going to talk about the fruiting body and also going to talk also the shape of the ascus itself, because the ascus also, they have different shape. It depends on the mechanism in which they are shooting the ascospores. So by this, I will stop my lecture here and I will continue with you after the break. Okay?
Thank you.
You're
Thank
welcome.
you.
You're welcome.
Thank you.
Take care, bye. Make use of your study week, huh?